So, what are you trying to do? I guess that's
that final $ map.
Yeah, so that's in the directory with all the files. It should be an RData file.
Well, but when I plotted .final.map, I got nothing, but we had generated that artificial data set.
Have you loaded in .final?
That's a good question. Well, I set my working directory to...
So it's part 3. Yeah, you have to load it. No, so it's an RData object.
Okay.
In the part 3 directory there. That final part 2. So you see that one there? If you load that RData object, if you look that second one.
Okay.
So you have to load that in, and then that final will exist. So I kept it separate files for the different parts.
Even though I set this as my working directory, part 3.
You still have to load stuff in.
Well, but so I just hit load and that didn't work?
No, so that's not the name of the file.
Oh, okay.
That's all.
I see what you're saying.
So here it said I couldn't find the GT plot. So
Yes, so Qt plot is not part of npmap.
Okay.
It's a separate package.
So I had to install that.
Yes, so if you look
I
on...
have to use this, or
You can't even use that. So
Okay,
if you look
that's
on
easy.
GitHub and search for npmap interactive,
Okay.
it'll come up and there's instructions on that website
Oh,
how to
okay.
install it.
Okay.
But it is complicated, so that's why I didn't go into it.
Similar than map, MP map interactive.
Right, so if you look for that on GitHub, you'll find it. Yeah. Okay, I can see people talking, but I'm going to get going again, just in the interest of getting through some of this. I put a few slides in here about mixed models, because you can fit that in MPIM. And it can be very important for including design information. So especially in Australia, we have a lot of variation between sites, even within sites. So we want to include that in our model of the phenotypes. And so it might be variation across rows or columns in a field trial. It might be variation between experimenters in milling or baking. And it might be variation over time. It might be relationships between individuals if you want to include a genomic relationship matrix. You can include all that, but the implementation in MPMAP requires an ASRML license. And so you have to know how to fit an ASRML model first. You have to know what mixed model you're going to fit. Once you've done that, you put it in as the base model. So that's the argument there. Basically, you fit your phenotypic model in ASRML first, put that in, and then MPIM takes care of adding on the genetic information. You can do this in one stage, so essentially you're putting all of the phenotypic information and the genotypic information in together, or you can do it in 2 stages where you fit the phenotypic model, pull out predicted means, and then you use those to fit the genotype model. That will be faster, but it's not as technically correct as the one stage. The other thing that's in there, which I haven't talked about much, is composite interval mapping. So there you're allowing additional covariates to be fit in the model. It's not as efficient as the implementation in Carl's package. Basically, you're doing a stepwise selection of covariates from the total number of markers, and you're trying to account for that background genetic information. NCOV, so that argument, NCOV, is the maximum number that will be selected. It's not necessarily the number that will be selected. If you put in a value of 1,000, it's most likely not going to include 1,000. What it will do is it will search, it will put in variables which are significant up to a point, and it will then start removing them. And if it only finds 10 which are significant, then it's going to stop. It's not going to try and fit 1,000. You should be aware it can be very slow when there are many markers. If you put in 10,000 markers and you do this, it's searching over all 10,000 multiple times to find the covariates. So it's going to take longer than your actual analysis will. So as I said, it's handy for smaller data sets, and you'll see an example of how it actually improves your localization of QTL profiles, but I would be cautious about using it if you have a very large data set. Then once you've fit whatever model you're doing for QTL mapping, there's another function called fit.mpqtl. You can just run it as fit. And what it does is it includes all the QTL in the same model. So the idea there is to try to account for any relationships between the different QTL you found. If you for some reason have picked up two which are right next to each other, then once you include both in the same model, one of them is not going to be significant, most likely. Typically, you're going to reduce the significance relative to fitting these individual models with the QTL. It also estimates the percent phenotypic variance explained by the whole model. Q-index is one of the parameters for this function, and that allows you to select out certain QTL to fit. So basically what it produces is a table of all the QTL. If you only want to look at a subset of them, then you just pick out the indices of that subset and it'll fit that model instead. So it does things very similar to what Carl was talking about yesterday after lunch in terms of selecting multiple QTL to fit in a model and looking at their effects. It just does it in a slightly different way. So if you run fit on those results that you produced for the Arabidopsis data set, then what it produces is a very similar table. So the dollar table at the end is important, otherwise you get out a huge list of information. The dollar table produces this table which looks very similar to the summary information. The main difference is going to be the p-value, and then the percent variance explained is also listed there at the end. The percent variance for each of the individual rows is how much is explained if you just fit that QTL in the model. The line which is printed out at the beginning is the percent variance explained by the full model. In this case, because you can see I called fit twice here, once fitting a subset of the values and once to actually run it and produce the full table and pick out that subset, what we see is two lines where the first one is explaining, okay, well, if we just run it on the full results that we found, we explain 62% of the variance, 63%. If we subset it to these ones where I've said, okay, in the full model, once we fit them, they have to have a certain significance threshold, 0.1, then we only explain 22% of the variance, and we only get 3 QTL out. So reducing the number of QTL that way obviously explains less of the variance. You'll note that adding up the percent variance of the 3 QTL does not give you the percent variance explained by fitting them all together. That's typical, particularly in this case because 2 of them are on chromosome 1. So they're kind of interacting with each other and they're not individually, they may explain more of the variance than once you put them together. If we compare the simultaneous model, the output from FIT, with the output from the summary, which is just fitting a model with 1 QTL in it, then down here I've just output the basically a subset of information from both of them. A lot of it overlaps, so the chromosome, the position, the flanking markers, and so on aren't going to change, but the p-values are. And so here, these p-values, this column is from fitting them all together, and the individual p-values over here on the right is fitting them separately. And you can see they can change, they can go both up and down, that is possible, but typically they get reduced once you fit them in the same model because there are other QTL which are affecting your estimates of their significance. I should also note the percent variance explained is computed from the adjusted R-squared from the linear model, which includes these QTL. We use the adjusted R-squared both because you might have a lot of QTL, but also because for each QTL, if you're fitting the full model, you have multiple founder effects. And for Arabidopsis, each of those is 18 different effects. And so you're adding in a huge number of variables that you need to adjust for in some way, otherwise you can quickly overfit the model. Yep.
Effect size and direction.
Yep. So as I said, for the Arabidopsis, it's not output because in this case, well, we didn't want to print out 18 effect sizes and standard errors. For the 4-way and 8-way populations, if you simulated data and the exercises has a simulated data set, I believe, with QTL, then it'll print it out as part of the summary. And they're stored, so the output, the MPQTL object stores all of the information. It stores the p-value information, it stores the test statistics, it stores all of the founder effects and standard errors added onto the object, so it can get quite large. So just be aware of that. It can often be better to just rerun the analysis if it's not going to take too long than to store all of that information. And yeah, yeah, I can show you exactly where. So here, so if you look at the names of MPQ full, there's a component now called QTL results, and that's a list of various things. And if we open it up, then we see it's basically storing a lot of the inputs you put in, but then it's also producing this p-value, walled, standard error, degrees of freedom, and the founder effects. And so when we do the summary of this, it pulls out those values for the specific QTL so that you don't have to go searching for them, but it is there for every location on the genome, actually. And so if you, for example, if you rerun find QTL and you produce a smaller set or a bigger set of QTL, it can go back and get that information for you. The last one there is actually essentially, that one should, oh, sorry. The last one, the QTL, is actually some summary information about the specific QTL. So you can see here what's being stored is a lot of information, so that's not going to be useful. For each QTL, it's a little bit obscure, but that's basically what's used to generate the summary table. So it's storing information about the position, about different effects, and so on. The other thing you can get from both the plots, you'll see the green bands on there, and there's actually a function to get these values directly called support interval, is to calculate the log drop-off from the peak of the QTL. And I think Carl talked about that yesterday. So this is relatively simple. You put in what log you'd like to drop off from the peak of the QTL. So here the log support is 2. And you call this function support interval, and what it prints out is for each QTL what the lower and upper bounds of that support interval are, and it also gives you the actual location of the QTL so you can see where it fits in the middle. By default, it's included in the plot of the QTL profile, which can get a little bit messy if there's a lot of QTL actually. And that's what it looks like in this case. So this was for the grouped model on chromosome 5. There's 3 peaks which are detected, and the support intervals are there. They're relatively narrow for the most part. As far as significance thresholds go, I haven't talked about that much. There are a number of ways to calculate it. So in our QTL, permutation is the preferred way. This does take a long time in MP-MAP, but it's possible. It's certainly possible. There are easier ways to do it. One is to do a Bonferroni correction, which tends to be conservative because you're dividing by the number of tests. You're dividing your you're multiplying your p-value by the number of tests you perform. So every location that you're testing at on the genome. You can also run empirical simulations. So you can generate data from your map, from your founder genotypes, even with your genotypic information, generate random traits and try and see how often you detect a p-value which is as significant as what you've detected in your observed data. That's relatively fast, but it still does require running analyses many times. There's a function called sim.sigthr, I believe, which will do this. Yeah, so basically it comes down to an issue of balancing the time and the computation versus how much power you want to have to detect things, or otherwise looking at the raw p-values and essentially figuring out, well, what's the most important thing? I mean, the p-values are meant to guide you in determining A, whether something is a real result, and B, whether it's important. But the founder effects are going to be important there, and the percent variance explained as well. So all of those will play into what you want to do with a QTL, and in any case, you're going to want to validate the QTL in some sense after you determine it. All of this is really meant to be an exploratory analysis to try and discover QTL. It's not going to tell you if they're real or not for sure. I've put in this image from a review paper that was published earlier this year, which is indicating some of the other models you can use. It's a little bit small, but hopefully on your screens you can read it. There are a lot of different ways of dealing with the QTL mapping problem in multiparental populations. As I mentioned before, you can look at markers or you can look at founder probability, so the full model or the biallelic. You can look at fixed effects or random effects. And there are a lot of different references here and in that paper, which was in TAG, which, if you're interested in looking at other approaches, then I highly recommend you look at these. It kind of depends. As far as finding large effect QTL, they're all going to perform pretty similarly. The differences will come in terms of finding smaller effect QTL and in terms of interpretability. So that's probably the main differences that you'll see, and in how you account for some of the variability in phenotypes that you have, how you deal with multiple traits, and so on. So depending on what kind of situation you are in with your traits, you may want to look at other different models. So there's references here for some of the things I mentioned, and then for this one, for this session, I really just generated one data set, SIM3, which should be in the directory, and there are some QTL which have been generated there, and basically the idea is just to play with some of the different models and see which ones tend to work better or worse and what kind of results you get out of it. Yes? Oh, okay. Any questions about QTL mapping? Okay. Okay. I'm going to leave you to work on the data set for a little bit, but first, in terms of guiding what we do after this, I want to get some votes on what people are interested in hearing about. So, where is my folder? Okay. There are a lot of different things that we can talk about, and you guys get to choose. So I've put 4 different options there. I mean, we can see how much time we have to talk about it, but who's interested in hearing about missing data imputation, in particular in the case of genotyping by sequencing? Okay, all right. I'm not counting, so I'm going to have to guess. Selective phenotyping. So if you have all of the genotype information on your population, how do you decide what you'd like to plant out if you only have a limited amount of resources? Okay. Who wants to hear a bit more about simulation and estimating recombination events? Okay. And then who would be interested in looking at a bit of the visualization capability? So I've shown you some plots, but if you want to look at something a bit more complicated. Okay. Okay. All right. Well, I'm definitely going to talk about imputation. There were a lot of people on that one. Okay, I've decided I'm going to start with imputation. I'm going to leave selective phenotyping till the end because that's actually relying on a separate package, whereas the other ones are using MP-map. And then I'll talk about simulation, recombination, and visualization, and we'll see how far we get. So imputation of missing data has become more of a problem, I think, recently because genotyping by sequencing is so appealing. You get a lot of data, but you often have quite a bit of missing data. But you can have a lot of different causes of missing data. So with GBS, it could be because your alignment to your reference is not great. It could be because it's poor quality or you don't have enough coverage. It could be, for example, when we deal with SNP data, it's because often our heterozygotes aren't called. It could be because you have different platforms. So you've collected data, for example, we've genotyped our populations using a 9K SNP chip, using a 90K SNP chip, using GBS, and we had different lines genotyped on each platform. So we have basically big chunks of data for our lines on different platforms, but they don't necessarily all match up. So you might want to impute your data. There's a lot of different approaches out there which were designed for association studies. So I've just listed some of them there. The issue with all of these is they require a very high density genotyped reference panel. The idea is you have some other reference panel from a population study. In humans, it's typically HapMap. You go through some phasing, and you're trying to impute that low-density targets. And for that, these approaches almost universally use information about pedigree or use information from a hidden Markov model. And then what you get out of it are probabilities, you get out phases of your genotypes, and you get the imputed value for your missing data. This has pretty good performance. So when I started looking into this, I was working with researchers at URI on a rice magic population, and they had GBS data on their parents and their progeny, and there was quite a bit of missing data. And we were looking at, okay, how well do these imputation approaches, first of all, how well do they work in magic populations? And furthermore, you know, is there anything we can use which doesn't necessarily rely on having a high-density reference panel? So here, the reference panel we were using is the parents. We are using those to infer what's happening in the progeny. And so this is results from simulations where we were looking at different marker densities, different sample sizes, and different percentages of missing data, ranging from 30% up to 50%. And then we were comparing different approaches. So B stands for beagle, which is very commonly used in livestock. M was just using the founder haplotypes to infer information in the progeny. And K was using K-means nearest neighbor imputation, which doesn't use a reference panel. As you can see, not using the reference panel reduces your performance. So, beagle does pretty well, but it's not using any of the information about the design population like the founder haplotypes are, and so you get a slight decrease in performance. It's not huge, but you go from about 96% down to 94%. But the question that came up was, what if the reference panel is incomplete? What if you have missing data in your founders as well, and that's affecting how you estimate your founder probabilities? I guess the simplest solution is to try and get more data, trying to get higher coverage, trying to use a different platform, replicates. But once you've collected the data, sometimes it's just not possible to do that. And so we were looking at what can we do to try and first impute the founders and then impute the progeny from that. And what we ended up doing is starting with the raw data, generating founder probabilities from that in spite of the missing data in the founders, and then essentially auditing the alleles that we were getting out of these founder allele probabilities to see how many of them. So for example, if we had founders A, B, C, and D, so we have A, B, C, and D, and we have some missing data here, so 0, 1, 0, so on. Some of the markers we have missing data, some of them we don't. We run our hidden Markov model, we get our founder probabilities out using the data, even with the missing values. And then for each of these markers, we audit the individuals that we have. So this is line 1, line 2, and so on. And we say, okay, this one, we're pretty sure it's inheriting alleles from founder A. So that may be based on the other markers. And we actually have an observed allele for this. So we know that this was a 1. Okay, so we've observed data for the progeny, and we're pretty sure it's coming from founder A. And if we look at this and it's not conflicting too much, we can be pretty confident that this should be a 1. So that's the basic idea. Once we do that, we can fill this in as much as we can, and then we can use it to re-estimate our founder probabilities and use that to infer the progeny. This actually performs very well in terms of filling in our reference panel. So these simulation results just for the founders. Here, again, we have varying marker density, we have varying sample size, missing data percentage. This is the percent of the founders which had data missing to. So this one down here, at the beginning, you only had 10% of the markers which were complete. So like this one, 10% of your founder markers were complete. All the rest had at least one missing value. And in that case, with the smallest sample size, we're able to get our founders after this reconstruction process back to nearly 100%. So this is over several hundred replicates. In all the other cases, we get a perfect reconstruction of the founders. So by using basically the two sources of information, the genotype data in the parents and the progeny and the pedigree information, we're able to reconstruct the founders completely and get back to the situation where we have this high density, high quality reference panel. And so then after that, of course, we go back to the situation where we get about 96% or more accuracy in our imputation. The last column there is again trying out the k nearest neighbors, and you can see it doesn't do quite as good a job. And then of course once you have that error in your founders, you're much more likely to have errors in your imputation of your progeny. So there's a data set simulated with missing data there in the part 4 folder. If you look at the founders, then that line of code is basically just saying how many missing values are there in the founders. I gather this was generated with 8 founders. And you can actually look at how much missing data there is in the progeny as well, which looks something like this. So this was generated with 50% missing data, 25% missing for the founder. So it's just looking randomly across the lines how much missing data there is. And the function we use to reconstruct this data is called mp-impute. So it has options for imputing the founders, for imputing the finals, or imputing both of them. And it does really rely on the founder probability calculation. So it does use that function mp-prob, which means that if you really want to, you can use either happy, you can use RQTL or something else to calculate those founder probabilities. And then essentially you just run on your data set. In this case, it actually imputed the founders completely correctly. So sometimes what happens, so it can impute it wrongly, of course. In this case, it's not that bad a situation, and it matches up with our previous simulations where we basically reconstructed everything correctly. In some cases, you end up with missing data still in the founders if you don't satisfy certain criterion when you're doing this auditing process. In this case, there was no missing data. All of them were fully imputed, and as it turns out, they were all correctly imputed as well. So this command here is just saying how many of the imputed founders don't match the originally generated data set. On the other hand, when we look at the imputed progeny, we don't do quite as well. So in this case, there's about 9% which don't match up, which is a little bit lower. It's on the low end of what we saw in simulations. Typically, we did see more like 96%. It does highly depend on your marker density and particularly on the size of your gaps. So that's partly why this method is really recommended for genotyping by sequencing. We have very, very dense data. We only ran simulations where at most the markers were 2 centimorgans apart. If you have things which are more like 5 centimorgans, 10 centimorgan gaps, you tend to get lower performance. And that's what we saw in the Rice data. So the performance in the Rice data was a little bit lower than the simulations, mainly because we had some gaps in the map which were 7 to 10 centimorgans. In practice, I would say rather than just running the imputation on your data and then using that straight away, you probably want to test it and see how much your marker spacing, your actual marker spacing and your sample size is going to affect it. So one way to do that is to mask out some percentage of your data, set it to missing even though you know what the values are, try the imputation and see how well you're recovering what's missing. Obviously, it's going to be affected by the marker density, by the sample size, by the type of genotyping platform, actually. In GBS, there's a lot of reasons why it might not be working, and so you might have larger regions where you're getting worse performance. And it could be affected by the level of heterozygosity as well. Okay, so that's the end of the imputation. Does anyone have questions? Okay. I'm going to skip ahead then to, where'd it go? Simulation. So I mentioned that just briefly there when talking about the imputation. It can be a useful tool in terms of just seeing how certain methods will perform on your data. So far, we've seen a general magic simulation, so sim.mpcross, where you can vary the map, you can vary the pedigree, and you can vary the QTL. With the QTL, you input the chromosome, the position, and the founder effects that you're interested in. I talked briefly about it with the missing data, but there's a lot of other things you can do with simulations. So I just said you can assess the imputation quality in your data. You can also compare different designs. This is something that I've looked into a little bit, trying to compare what the effects of different numbers of generations of advanced intercrossing is in your population. I'm currently looking at the effects of using double haplines versus RILs, and people are certainly interested in how you compare MAGICs versus NAM populations as well as different forms of MAGIC populations. You can also use it for QTL mapping to test the power for different approaches, see which ones might work best. You can generate empirical significance thresholds, estimate power for your specific data, so for your map with your genotype data, with your founders, or some combination of those, see how likely you're going to be to detect QTL of various sizes, and you can see how theory basically compares to reality. So in terms of comparing designs, the main thing is to generate different pedigrees. And then you generate data from them, and then you compare whatever you want to compare. So typically you're comparing the number of recombinations, the size of the haplotype blocks, you might be comparing the extent of LD, there's lots of different things. For the most part, it's about genome structure, but if you want to go further, you can actually do QTL mapping in them and see which designs tend to have more power. So down here are some examples of different types of pedigrees you can generate. The main function is called sim.mpped, and there are a number of parameters there. As I said, I mentioned it briefly. You can put in the number of founders, the number of funnels, the number of individuals to generate. You can even put in the number of seeds to be pulling out of each line after you get to the intercross stage before you go into selfing. And you can change the number of generations of selfing as well. So you can do that for the 4-parent, the 8-parent. This third one here, this IRIP-gen argument, is to put in additional generations of intercrossing after you create 4-way or 8-way lines, but before selfing. And this other function is actually in a different library called spclust, but it will generate a NAM pedigree as well, with a certain number of founders under the reference design and a certain number of individuals in each of the populations. If you want to set up other pedigrees, it takes a little bit more work typically, but there's a fairly wide range of things that you can generate just using these functions.